Okay, a couple of announcements to start. So the exam is on Friday. Please make sure that you've done the lockdown browser in my EduCat page and to test that it's working on your computer. You might have to do an update after a full summer of not doing that. So please make sure to take time to do that and don't do it like right before the exam. Like do it today. If you haven't done so already, make sure that you have a fully charged battery. And so I'll try to get here as close to 10 to noon as possible. And then the exam will open at noon and you'll be able to get into it on my EduCat page. There'll be two parts to it. There'll be the multiple choice portion and then the essay portion. I split it out because that way I can release the multiple choice scores as soon as everyone's done taking it, whereas the essay I have to, grade. So those get delayed, but at least you get the multiple choice feedback fairly quickly once everyone's taken the exam. So that means when you're in here on Friday, whatever one you start with, you have to, I believe you're going to have to exit all the way out of lockdown browser and come back in to do the next one. Otherwise, there's glitchiness. So let's try that. Just let's say you choose to do the multiple choice. What you should do is raise your hand when you're done with whatever portion you tackle first. Let's say it's the multiple choice, raise your hand, show me or the assistant that that you've submitted it. So we know that you've done that. Then you'll have to get out of lockdown browser and come back in almost like you're, sitting in a new class and taking a new exam. Just pretend like it's like it is like that. And then show us the second one. If you take the essay, the second option, show that that has been completed before you leave the classroom. Okay. Make sure to ask questions, raise your hand, and I will be circulating through to answer any questions. And I really, really encourage that. You have to appreciate, I'm me, you're all of you. We all have different brains. So the way I might phrase a question when I'm typing it up, it may, to my mind, it's like, oh, that's full of clarity. But to your brain, you might be going, what? So I'm not trying to do any kind of trick questions. It's just your brain might not click with how I phrased it. So then raise your hand and I will try to rephrase, make a comment that might jog your memory. Okay. So take advantage of me being in this room with you if you have any questions. Also, so today's Wednesday. That means the lab quiz option has opened up for you to take the lab quiz. And so you have 24-hour window so you can take it now. No, not now, don't do it right now, but you can take it right after lecture if you want. But it closes, you only have a 24-hour period. So if you are in the 9 a.m. lab, it's gonna close right before that. So make sure you do find a time today. And same with the 1 p.m. lab, it'll close right before 1 tomorrow. So please make sure to do that. Once you start the lab quiz, the clock starts ticking. You have 20 minutes. Okay. Questions on like the process of the exam or the quiz or anything like that? All right, I will have a short lecture today and allow time for questions and clarifications that you might need on the exam content. I won't stand up here and lecture. I'm open for questions. All right, so if you need something to be clarified, ask questions. All right. Oh, the other thing regarding Friday when you're in here, I'm okay if you want to take out a blank piece of scrap paper. It has to be completely blank, obviously, but sometimes that helps students when they're trying to think through something, if they can scribble or draw instead of, your only option is when you actually do your answers to type. And So if you want to have that piece of scrap paper on Friday, I'm okay with that. And if that helps you, great. And then just remember to show that you submitted both the multiple choice and the short answer portions of the exam before you leave. Okay. So I'm going to lecture for a little bit, and then we will have time for exam review and questions. So I think we left off here. So we're talking about Phloem translocation, which will not be on the exam. And this is the idea of when they were trying to figure this all out. This kind of questioning started back in the 1600s when they were trying to figure out how does sugar get transported throughout the plant body. And so it was a long, long process of many, many scientists getting into this. And, of course, they had to figure out the phloem tissue and cells. And so here you're seeing the idea of the phloem again. We've seen this already. And so in angiosperms, we talk about the sieve tube elements. So that's what you're seeing here, the sieve tube elements and their associated companion cell. So you see here that there's several sieve tube elements that are fit together end to end. They just stack on top of each other, so to speak, and then each one has an associated companion cell with it. The area where they attach to each other or butt up to each other is called the sieve plate area because it has these pores, but they're open. There's no membrane crossing there. It's just an open channel that allows for flow from one cell to the next. They're living cells, so they don't have that thick secondary cell wall. You can see that the artist has drawn some of the organelles in there. And so they have some organelles, they've lost some organelles. And so they're not fully up and running all on their own. That's why they need the help of the companion cell. And so often you will see, and the artist has drawn several connections between the companion cell and the sieve tube element. What are those tunnel connections called? Plasmodesmata. And so you'll often see that when companion cells, they have lots of plasmodesmata to feed, make sure that the associated sieve tube element is getting all the lovely nutrients that it needs to stay alive. There's, cytoplasm in here. So they had to think about all of this, like the impact of the frictional forces of the wall of the sieve tube element, all the cytoplasm, the organelles. And so they had to think about all of this once they discovered the actual composition of the phloem tissue. And so just keep in mind that those sieve plate areas where they connect one, end to end, those are all open. There's just a tunnel system through there, okay? It's open channel or tunnel. These pictures are just trying to show that the companion cells are right there. There's different kinds of companion cells. They give them all kinds of different names. We're not going to get into that. But in general, there's lots of plasmodesmata that connect the companion cell and the sieve tube element. And then this table is just looking at sieve tube elements and some of their characteristics. And so the open channels, they're open. We're going to talk about P protein and the fact that the companion cells are helping metabolically. They help out the sieve tube element. Okay. So when they were looking at the sieve tube elements and studying their internal composition, one of the things they did notice was the structure or this protein that they nicknamed or named the P protein. And then they also discovered something else called callus that can be often present in sieve tube elements. And so sometimes it's there, sometimes it's not. And so they started wondering what is going on with this. And so far anyways, they don't see it showing up in gymnosperms, the P protein. It seems like only angiosperms have this mechanism that they employ under certain conditions. And so when they analyze the actual amino acid content of the protein, it varies. So it's not identical species to species, but it seems to have a similar function. So it's like the different species of angiosperms have, come up with their own version of this. And it seems to provide protection against injury. So if that very precious sieve tube element gets damaged, like maybe a bunny rabbit comes along and starts chewing on the plant and the sieve tube element gets damaged, what happens is the pea protein gets synthesized very, very quickly, and it actually will go to the puncture area of the sieve tube element where it's damaged and kind of block it up, seal it. And so they're just trying to keep maintaining the integrity of the sieve tube element so that there's not any holes in it from damage or herbivory or anything like that. Did you have a question? Yes, and how like our immune system and blood vessels, they'll try to coagulate, and yep, very similar to that idea. And so it makes sense. Often you'll see the pea protein tends to also accumulate in the sieve plate, when they look at it under the microscope. So it's at injury sites, and then some of it will be around the sieve plate until they can do full repairs. And so it is kind of like this temporary, quick, let's plug up any damage, and then they can fix the damage and move on. And so the big payoff, if they can do this quickly, is that we're going to find out that there's hydrostatic pressure inside the phloem tissue. And you want to maintain that in order for the phloem to do its job correctly. And we'll talk more about that. The callose is not a protein. It's actually a carbohydrate-based molecule. And it is also synthesized very rapidly in response to injury. And again, when they look at it under the microscope, they can see that the sidewall areas, if there's any injury, like especially around some of the lateral pores of the sieve tube element, you might see the callose starting to build up, or by the sieve plates. This one tends to persist longer. It seems to be a longer term solution compared to the pea proteins, so that when they experimentally do this, they see the callose sticking around longer than the pea protein. So just a couple of different mechanisms that they've discovered that help the plant maintain the physical integrity of the phloem cells, and to make sure that you can keep having that hydrostatic pressure in the sieve tube elements. Of course, remember, because the vascular tissue is all clustered, if there's like so really severe injury to a sieve tube element, they can reroute. We've talked about that with the water transport. There is that ability to reroute in the xylem. We're going to see it's the same idea with the phloem. They can reroute the transport if a cell gets so damaged. So here you're just seeing the sieve plate on the left, and then on the right you're seeing the buildup of callose after they experimentally injured that area. Okay, so they're starting to, decipher what's going on with what kind of tissue makes up the phloem, what's inside it. They're living. They don't have that secondary cell wall. They can make different molecules to help protect their physical structure of the cell. Then they started looking at, okay, well, what seems to be moving through the phloem? And the water and minerals, they discovered, that's the xylem's job, but what is the phloem doing? And so they had to experimentally figure out a way to measure the contents of the phloem tissue. But back then, the technology was very limited. And so actually extracting the phloem tissue without contaminating it with nearby cells stuff was a challenge. So some brilliant scientists came up with the idea, hey, why don't we use aphids? And aphids are little tiny insects with these very precision mouth parts called stylets that act like a needle, and they can puncture right into the phloem, and just the phloem. And then what they do, I can admire aphids, but I really dislike them because they're present in the greenhouse on a regular basis. But they're so good at just piercing the sieve tube element and then sucking the nutrients out of that. And then it actually goes, they use some of it for the nutrition. And they excrete out whatever's, their waste. They actually call that droplet honeydew. I mean, such a nice name for such a nasty insect. So they call it honeydew. So the brilliant scientists way back in the day said, why don't we release the aphids onto the plant, let them get going, and then we can collect the honeydew and analyze the contents in there. And so that's what they did. This lower picture is showing the mouth stylet, and it's piercing right into the sieve tube element. Precision feeding, really. Here's another from a published paper where they're showing the idea of the mouth part, the stylet, coming in to the phloem. Super accurate. And then, so the first set of experiments, they collected the honeydew, and then they analyzed what's in it. So being a scientist and thinking about this, what's going on with the insect, can you think of maybe some problems with this experimental design? Great. So the insect is that's food for the insect. Just like when we take in food, if we eat a hamburger, we don't poop out a hamburger, right? I mean, it gets changed, it gets altered. So yes, they can tell at least somewhat what's going on, but it's been altered pretty seriously by, of course, the insect digesting that whatever's in the phloem. So it was a good start, a good thought. So then they came along later and they said, well, why don't we release the aphids onto the plant, let them get their stylet in and start doing that precision feeding, and then let's anesthetize them and cut off their stylet. So that's what they did in this one. So they got it feeding, and then here's the scalpel coming in, and they're cutting the stylet off the aphid. And then it's still, in the sieve tube element, and then the drops start oozing out, and they collect it right from there. And so it's only passing through the stylet, not the digestive tract of the insect. Now, I thought that was pretty brilliant, actually. So here they collect those drops. That's pretty much not been modified. The stylet it's just a tube. And so that experiment gave them some more reliable data on what might be in the phloem. So this is what they came up with. And this is from castor bean. But you can, in general, state that the dominant substance in any plant will be sugars, hopefully, if everything's going okay. But you can see that there's a range of other molecules present as well, but sugars win, all right? So that's why we tend to focus on that the phloem is responsible for sugar transport, but you can see that there are commonly other organic acids, amino acids, proteins, minerals. Okay. So sugars, that's a little vague. There's all kinds of sugars out there, right? So it's like, okay, well, what is the sugar? And so they had to narrow that down. And what they discovered is that it's sucrose, which is being shown if you look at these two. So sucrose, take this bar and drop it down. That's sucrose. Sucrose is glucose and fructose bonded together. That's what makes sucrose. And then if you add a galactose to that, then you have raffinose. If you add another galactose, you have stachyose. And if you add another galactose, you have ribascose. Okay, so just depends on how many galactose you're adding. So I'll come back to this idea, but sucrose is the dominant sugar in the phloem. Okay, and I need you to know, not for the exam on Friday, but the next one, I need you to know that it's glucose and fructose bonded together. All right. So it's a disaccharide, two sugars, glucose and fructose. And then depending on how many galactose units you put on, you get those variety of other sugars. Okay. And so sucrose dominates in terms of the type of sugar, but you can find these other sugars in the phloem. Sugar alcohols can be in the phloem as well. Mannitol. Anytime you see OL on the end of a science word, that means it's an alcohol of some sort. So mannitol is a sugar alcohol. So sometimes you will see that in the phloem. Here's some example of amino acids. Glutamic acid or glutamate, glutamine. So those are important amino acids for the plant. Here are some uriides. And so those plants that can form partnerships with the nitrogen fixing bacteria, often you'll see these types of uriides floating around in the phloem because of the action of the bacteria. We'll talk more about that later. But let's zero in on sucrose. And so, of course, they like, oh, okay, why is the plant preferentially allowing for sucrose transport when there's all these other sugars? Why not just glucose, for example? Why not just do that? And so they had to think a little bit about that. And it's a disaccharide, so it's relatively small, so it's easier just physically to get circulating through the plant rather than a big, carbohydrate. It's also called a non-reducing sugar, which basically the upshot of that is that it is non-reactive. So it won't react with a bunch of stuff as it's flowing through the plant body. It just stays as sucrose. It doesn't like to react with other things. So it's more stable, less likely to react with other molecules nearby that it's flowing past. And then when you break the bond between the glucose and the fructose, there's a decent free energy of hydrolysis when you do that. And so there is a payoff of some energy for the plant when that bond is broken. And just its small size makes it highly mobile. And so those are all good ideas, good explanations for why sucrose seems to be the dominant sugar. Okay, here you're just seeing the idea of reducing sugars, which are more reactive. So a reducing sugar is more reactive. And so if you're a chemist, that will make more sense to you, perhaps if you see these figures, to convince yourself that the aldehyde and ketone groups are more reactive. And so you don't have that with the sucrose. Okay. So then they started looking at how is this moving through the plant body, through the phloem? What is the mechanism? What's the process? And ultimately, it came down to this idea that sugar moves from a source to a sink in the plant body. What does that mean? If you are thinking about the source portion of the plant body, that's a part of the plant body that is considered a net exporter of sugar. So that means it's producing so much sugar that it has its own needs met, plus it can send the extra somewhere else. So it's a net exporter. So that is typically leaves, right? The typical plant, it's the leaves that would be an excellent example of a source. They're busy doing photosynthesis. They make so much sugar that they can take care of their own leaf tissue as well as sending extra out to the rest of the body of the plant. So pretty much anywhere that's actively photosynthesizing, and mature leaves is probably a more accurate thing to think about rather than really young leaves, because actually really young leaves can actually be a hog for sugar. Like they need extra sugar, they can't make enough. So mature leaves are an excellent example of a source. A sink is the opposite. It's the idea that it's some part of the plant body that either can't do photosynthesis at all, or only can do some, but not enough to fulfill its needs. And so it's a net importer or a net consumer of the sugar. So an excellent example of that would be, a part of the plant body that just really never sees sunlight. And so you're thinking about the roots, right? So roots, but stem tissue can't do it fast enough or enough. Any young developing organ, really young leaves, young flowers, young fruit, all of that you can imagine where they don't have enough of the photosynthetic machinery up and running where they can do it all on their own. They need help. So you have to remember that sink tissue, just like source tissue, has to carry out respiration. They have to do all of that. And so there's energy that's required there. They have to have that sugar, extra sugar, to build more cells. If you think about a developing potato tuber, for example, to build that lovely potato tuber that you eventually eat, you gotta, of course, go through a lot of cell reproduction to build it. And then to store, again, the potato tuber is an excellent example of storage of sugars. You get it nicely stored up for over winter. So source versus sink. So then they say that sugar will move from source to sink in the plant body. And remember, in the phloem, it can go all sorts of directions. It can go from a leaf down to the root. It could go from a leaf on this side of the tree over to a leaf on this side of the tree. So it can go lateral, it can go upward. So often mature leaves, you know, they're lower on the plant. So then the mature leaves can send up sugar to a young leaf. So it's pretty much any direction you can imagine. So PS is photosynthate. So often you'll see that word. Photosynthate is primarily sugar, but they use that word because they understand it's not always just sucrose. There's, you know, raffinose, stachyose. There's other options too, but so you might see that word quite a bit. So translocation just means movement of photosynthate or sugar from source to sink. And it really does depend on how close are the source and sink to each other. Sometimes they're really close and the travel is very short. Other times it's a significant difference. It highly depends on what is the plant doing in its life cycle. Is it a seed that's just germinating? Is it a mature plant that's going into the fall and needing to, you know, drop its leaves eventually? And so it just depends on its life cycle, how this is going to impact the movement. Certainly the vascular connections, how direct are they? Faster, if they're super direct with each other, it's going to be an efficient flow. And then they did a lot of experiments with pruning as a way to try to figure out what's going on with the phloem. And so pruning is just, you know, cutting away parts of the plant body and seeing what happens to the flow of photosynthate once you do that. And we'll talk a little bit more about that later. So they are, you know, trying to piece together this whole process, and they're trying to figure out, you know, they eventually did figure out that it can be pretty much any direction of movement through the sieve tube elements. Here they're doing some carbon dioxide, radioactive carbon dioxide that they labeled, and then they exposed the plant to that labeled CO2. The plant took it in, made sugars from it. And so here you're looking down on the plant, and so the younger leaves are like on the very top of the plant. And what they're just trying to show here is the darker colored leaves are getting more of the sugar than the light colored leaves. And so they fed it to this leaf, and they bagged this leaf and put the CO2 in the bag, and the leaf took it up, and then it gets translocated. The sugars get translocated to the very young leaves, is getting a lot of it. Okay, it's dark green, but so are some of the leaves that are, so 1 and 6 actually are getting more because they're more directly in line with that leaf they treated. So they were just looking at how direct, you know, the connections are. Do they get more? The answer is yes, if it's really direct. A little bit more of a lateral movement. They still get sugar, but it's a little bit less. And so they were playing with all of this and trying to figure out how that impacts translocation. Here they're just showing the sieve tube elements, and they were running experiments to demonstrate that you don't have any membranes crossing sieve plates. It's all open. And so they were slowly, over a couple hundred years, piecing this all together. And they came up with the key scientist later, you know, a few hundred years after it started, it was Dr. Munch. And so you might hear about the Munch Phloem model. And so he was the one who started to really come up with a hypothesis that they could test, and it turns out he was correct. And so, but they had to factor in, you know, what is the structure of the sieve tube element? It is living, so you got to think about cytoplasm and organelles and all the frictional forces. They could measure the actual rate of translocation, and so they knew the rate. And so whatever hypothesis they came up with had to satisfy what their direct measurements were showing. They knew that it could move in different directions through the phloem tissue, so they knew that. And then they started thinking about, okay, the leaf cells, like spongy mesophyll and palisade cells, they're the ones doing all the photosynthesis. And you got to get the sugar from those cells into the vascular tissue. So you have to have, they had to figure out all that, thinking about how's that happening, and we'll talk about that in a bit. And then, so all those spongy and palisade cells, they're all doing photosynthesis, but then they have to direct the sugar into the vascular tissue and get the vascular tissue the sugar so they can start moving it through the plant body. So getting that photosynthate then into the sieve tube element in the phloem tissue, you know, and getting it sent through the plant body. And then once it gets to its destination, you got to unload it into the tissue that needs it. So if you think about maybe a potato tuber starting to be developed, you got to get it out of the vein, you know, out of the vascular tissue and get it into the actual cells of the potato tuber. So that's the unloading. So that getting it from the leaf tissue into the sieve tube element, they call it loading. You're loading the sugar into the vascular tissue, the sieve tube element. And then you got to unload it once it reaches its destination. So they had to figure out all of this. And they came up with a hypothesis that they call a pressure flow model. So pressure flow, we're talking about a pressure gradient again. But this time, it's a positive pressure we're going to be thinking about. Not like in the xylem, where we said it's actually a negative pressure that is allowing for water to move from the roots to the leaves. So now we're still thinking about a bulk flow process, the delta P over delta X idea that the Poisson scientists came up with, the bulk flow concept. And it's due to water pressure, so it's hydrostatic pressure, and it's a gradient. So what we see is that you have to have a gradient between the source and the sink. You have to maintain a gradient for there to be flow. If the gradient dissipates, then the flow stops. Okay, so what we're doing is thinking about this movement of sugar from source to sink. And it's actually the condition is that the source builds up a hydrostatic pressure. The sink has a lower hydrostatic pressure, and so you have that gradient from higher pressure in the source to lower pressures, maybe to the potato tuber. Okay, from source to sink. And it's very much linked to what's happening to water movement in the xylem. So we're going to see that a lot of the water that's going to rush into the phloem is actually coming from the xylem to allow for that hydrostatic pressure to build up. So in visual format, this is what we're thinking. So this is just showing what's happening in a source. So let's say it's a mature leaf, and you can see here the sieve tube element and the companion cell. And so what you have to imagine is there's nearby, let's say there's nearby spongy mesophyll cells here. And they're doing photosynthesis, and then they give all that sugar, which is the red balls, they give all the red balls, the sugar, to the companion cell sieve tube element. Well, you have to think about water potential concepts here. If they start loading all that sugar into the phloem right by, you know, the leaf cells here, you get more and more sugar building up in the phloem of, you know, the vein of the leaf. That's going to mess with your psi sub S because you're putting more and more solutes, So what will happen to your psi sub S value? Gets more negative. So I prefer that statement. It gets more negative. Okay, that's a really accurate way of saying it. So psi sub S becomes more negative as more and more sucrose gets dumped in there, gets loaded in there. Okay, so then that will make you think immediately about psi sub P. If you have a bunch of solutes getting loaded in here, it's driving your psi sub S more negative, which is driving your overall water potential more negative. What's going to start to happen? Water's going to flow from less negative to more negative into that phloem that's getting all that lovely sugar. So now your pressure is starting to build. Hydrostatic pressure is starting to build. So you set up the situation now in the leaf where you're generating that nice hydrostatic pressure. So that's only half the story. That's just half. You got to finish the thought, finish the story. So let's look at a sink. Let's say it's the potato tuber, and you have the vascular tissue coming down to the, well, actually it's an underground stem. Potato tuber is actually a modified stem. So you have the vascular tissue going to that underground stem, and you have the situation where that sugar now is getting unloaded into the potato. So as you take sugar out and unload it into the potato, what's happening to your size of S right here then? You're getting rid of sugar, it's getting out of there. What's happening to your size of F? Getting less negative now. Which means you're going to create a situation here where it's going to be less negative than somewhere nearby. So water is going to start to exit. It's going to go from less negative in the phloem to often it goes right over to the xylem. It's more negative. And so what you've done now in this situation is in the sink tissue near that potato tuber, that phloem has dropped its pressure. So you had high pressure being generated in the source, lower pressure now in the sink. So now you have a pressure gradient. And as long as that pressure gradient is maintained, you'll have flow of sugar that's dissolved in water coming from the source to the sink. But eventually that potato tuber is going to get to its mature size. And then it's going to say, okay, I'm done. I'm mature. I'm good to go. And so that gradient can then dissipate. And then another tuber maybe nearby is going, I'll take that. I need it. I'm still growing. All right. And so you can get a switch then. So eventually you'll have other sinks going, I want my fair share. And so then you'll get transport to those. Okay. So it's very dynamic. Just depends on what's happening with the plant. Questions right now on that idea? So I'm going to ask you to do an odd thing and compartmentalize this knowledge I just gave you. Put it in a box, set it aside. Please don't confuse that with what is going to be on the exam on Friday. Because in the xylem, we don't have this idea. Okay? So compartmentalize it and just let it sit. But now we need to stop. I'll stop here with the lecture. Let's think about the exam. And let me pull up some of the slides so that we can use that as a way to facilitate any questions. So I'm going to start with the first slide set. Plant and cell architecture, right? Yeah. So and let me share so that people can see it. Okay. Why is it doing that now? I swear my laptop is possessed. I have to go get this checked. It is just ridiculous. Oh, it's still. How about this? Okay. All right. So, plant and cell architecture. I asked you to learn the basics of the plant body. Remember, there's assigned textbook readings as well with each of these topics. Make sure to pull up that Word document as you do any of the readings because it tells you exactly what page range is. I don't think I've ever asked any of the chapters to just do the whole thing. There's always like only certain sections. So to be efficient, pull that Word document up to read through what you need to read and study. Questions on plant and cell architecture? good question. No, I won't ask you percentages. Primary cell wall, you know, we tend to focus on cellulose quite a bit. So knowing the general composition, so the words, but not the numbers. Yes. So this one, the one on the fibrils, microfibrils, that one is important. Like these two slides, understanding kind of the three-dimensional aspects of what's going on with the cell wall. Certainly knowing, you know, primary, all cells have that, but then you can have secondary, and what does that mean? What's the composition? What's the consequences? In general, yes, knowing the organelles of a plant cell, and then what do you think are probably some of the more important organelles that you might see more questions on? Yep, vacuole, chloroplast, mitochondria, for sure. And, you know, especially you don't know this yet because you haven't experienced it, but you know, the chloroplast is never really going away. We're going to talk about that for a long time. So, yes. Okay, good question. What's going to happen if I do this? Let me pull that up because it's a little, I've had some confusion on this in the past. So looking at the chloroplast in general, you have two membranes surrounding it. Okay, so there's the so-called outer membrane, so-called inner membrane right underneath it. But then there's a third internal membrane, internal, floating around. All right, it's just floating around in the cytoplasm. Okay, so if I were pointing at this whole thing and I would say, oh, look at that thylakoid. Look at that thylakoid. Isn't that pretty? All right, so when I say that, I'm referring to the entire internal membrane, the whole thing. You just use the word thylakoid if you're thinking about the entire thing. But you can tell that there's different parts of it. I mean, it's not all homogeneous. So there's the stacked region, the relatively unstacked region. So they had to give names for those too, because sometimes we do have to zero in that specifically. So if you're talking about the stacked region, that's the granum or grana for plural. That means if, so I would say, oh, look at all those grana. Then I'm just zooming in that region. There's several being shown here, so I pluralize the grana. If I said, look at that stromal lamella. Aren't those interesting? What I'm zeroing in on is the relatively unstacked, like bridge-like portion that connects the grana together. And, you know, I know it just seems like, you know, biologists are torturing us to make us learn more terms, but it becomes necessary because when we get into the light reactions, I have to use these terms. I have to talk about grana, I have to talk about stromal lamellae, so we do have to get that detail.
So what is the name of like individual bits? If they're not thylakoid.
The whole entire internal membrane is a thylakoid.
Right, like in the disc.
Each disc, I don't, it's just grana. Okay. Yeah. I don't know that they have a sub name for each like portion. I don't think they do. I've never seen. Oh yes, don't forget that. So the inside, do they have it? Yeah. Where's the word lumen? Right above D. Oh, lumen. Thank you. Good. So okay, the lumen. So if you do surgery, on the thylakoid membrane, and then you peer inside, look inside, that's the lumen. Just like your intestinal tract, the inside is called the lumen. The same concept. And yes, you need to know that because yes, later in the semester, I'm going to talk about what's happening in the lumen, and then the watery portion is called the stroma. So certain biochemical reactions happen in the lumen, others take place in the stroma. So you have to know the details for sure of the anatomy.
And we have to know how proton.
Not yet, but eventually, yes. Eventually we'll get into this whole concept that they're alluding to, but not yet. Friday is just anatomy. Let's get the anatomy down. Then we'll worry about the other stuff later. But this isn't going away either. This is forever in this semester. So obviously it's important.
And then like on the leaf cross section.
So the leaf cross section, again, very important, right? That's not going away either. So making sure you know the detailed anatomy for sure. So the, by the way, so they have to throw you all these extra words, but mesophyll is really, like, some people will tell you this is a palisade mesophyll cell. This is a spongy mesophyll cell. I tend not to say that because truly it's parenchyma. It's parenchyma. So I tend to say palisade parenchyma cell, spongy parenchyma, because it's a type of parenchyma. All right. So I tend not to use that mesophyll. But knowing palisade versus spongy, understanding that there's air, open air gaps in here, the vascular tissue, epidermis, cuticle. Something else I didn't mention, but it's really, really important. What else on there? Guard cells, the stomata. So if you think about guard cells in the pore, it's the stomata. Yeah, almost without exception. I can't really think of an exception. They will always, the xylem's always bigger. Really massive cell wall area compared to, so this is a root, and so that pus area is all xylem, very massive cell wall, very big cell, and then the phloem is these little delicate cells right in, and they usually are kind of blue-green, at least to my eye, and then the xylem is usually stained red. It's just a staining procedure that they do. What about water potential? Are we doing okay on that? So today's Wednesday. If you're having questions on water potential, are there any right now? Does anyone have anything I can quickly answer? Yes. No, the lab quiz will focus on the lab experiment, the potato stuff. So what you went through in lab, looking at percent change in length or weight, and then the Tartakov, and thinking about the concepts of, if it weighed more at the end, what does that mean? And how did you figure out your size of S? And so working through the actual experiment, like the concepts of what did you get from that, and how did you end up calculating your water potential? So those types, and the water potential ideas certainly come into play, obviously, with that lab experiment. So knowing the overall water potential equation for the lab quiz will be important, as well as the exam, of course. And by the way, remember the water potential. So psi sub W equals, what are the two components? Psi sub W equals psi sub P plus psi sub S. It's not a negative sign. Okay, it's plus. Even though psi sub S is negative, but you have to add them to, add it together. So if you are finalizing your studying on the water potential between now and Friday, and if you have a question, I check my email even after hours as we lead up to an exam in case there's any last minute questions that I can answer via email. Okay, or you can set up an appointment during the day to meet with me. Okay. All right. See you on Friday.
Are we all-
I think I have hours. I have